describe you all hepatitis viruses, what we have to talk about during the classes, and about main important things about the hepatitis viruses. So, of course, you know, viral hepatitis, it's a diverse group of microorganisms, so there are many different kind of hepatitis which can infect liver cells. So let's start. Hepatitis viruses include at least 6 viruses, but also we have several more groups which also widely described and medical important, but we won't talk about it because it's just well, time consuming, and in infectious diseases not so important that few what we will discuss about. So target for all of these viruses is the liver, and basically hepatocytes. Symptoms mainly are similar. They differ greatly in their structure, mode of replication, mode of transmission, and the time course and sequel of the disease they cause. So acute stage of all these hepatitis are primarily similar, but other things can be extremely different. Hepatitis A virus, shortly we call HAV. Hepatitis B virus, HBV. There are classical hepatitis viruses, and we will discuss about them a lot. Also, hepatitis C, G, A, E, and hepatitis D virus also will be described here, but shortly. Of course, much shortly than previous two. What's the reason to describe hepatitis A and hepatitis B very careful, instead to describe all of them? Because hepatitis A and hepatitis B virus has very different kind of structure, absolutely different ways of transmission, and mode of replication also totally different. But other hepatitis can be compared with some of them. For example, hepatitis C, D, and G can be put it in the group of hepatitis B virus by the mode of transmission. Hepatitis E, similar to hepatitis A. About that, we will talk. So don't scare about these strange apps like you can see here, hepatitis, non-hepatitis A and hepatitis B virus, like, I'm sorry. That one. There is nothing special. It's just non-A, non-B hepatitis viruses. The viruses because there are many, but not A, non-B. So let's describe. Shortly, I will introduce you all hepatitis, and then we will talk specially about that. So first, hepatitis A, or also known as infectious hepatitis, or in Russian books actually and articles, a lot of them are described as болезнь Боткина, like Botkin's disease. It's caused by epicoronavirus. We already know what does it mean, epicoronavirus. Last lecture we described that. Pico means small RNA virus. Is spread by the fecal oral route, has an incubation period of approximately 1 month, after which symptoms start abruptly. So abrupt onset, it's one of the characteristics of it. Does not cause chronic liver disease, rarely cause fatal disease. So as you see, hepatitis A virus can be easily transmitted through the fecal oral route. because the virus is stable for environmental factors, but it's one of the most non-danger hepatitis because never produce chronic liver disease. Okay, not never, I would say not usual, and very rare cause fatal disease. Another one, hepatitis B, serum hepatitis, we also call it, because it's transmitted through the blood particles, caused by a hepatinovirus, which only DNA virus of today's topic, is spread parenterally by blood or needles, sexual contacts, or from mother to child, has a median incubation period approximately 3 months, after which symptoms start insidiously, so not totally abrupt. It's followed by the chronic hepatitis in up to 10%. I found in some books, I found 20% of cases. We will say there are many, a lot of. Occasionally associated with the primary hepatocellular carcinoma, so it's like a liver cancer, but not in the primary infection, in the beginning of infection, of course, much later. More than 1/3 of the world's population has been infected with HBV, resulting in 1 to 2 million deaths per year. So can you see, more than 1/3 of people over the world infected by HBV? Very dangerous, I would say, situation. So there is a liver, picture of the liver, which described as cirrhosis and caused by HBV. You know, cirrhosis can be caused by many kind of factors. One of them, hepatitis B virus. Hepatitis C virus, also widely distributed with more than 170 million carriers of the disease, and a lot of them are chronic. It's spread by the same way as hepatitis B virus, but it's more prone to cause chronic diseases, I said. Up to 70% of all cases developing to the chronic infection. C virus also increased risk for hepatocellular carcinoma. C virus is flavivirus with RNA genome. So G, also flavivirus like hepatitis C and cause chronic infections, also as hepatitis C, and transmitted totally by the same way as hepatitis C, very similar to hepatitis C virus also. E virus, hepatitis E, is an enteric encapsulated virus with an RNA genome. It's only one of its family, and the disease resembles HIV. So hepatitis A and hepatitis E virus, very similar by transmission way, by the structure and nucleic acids. The last one, delta hepatitis or hepatitis D virus, is a unique in that requires actively replicating HBV as a helper virus and occurs only in patients who have active hepatitis B infection. Hepatitis B virus provides an envelope for hepatitis D virus, RNA and its antigens. Hepatitis D virus exacerbate and the symptoms caused by HBV. So such kind of virus can't replicate itself without hepatitis B, totally can't. So D virus need B virus for replication, and D virus decrease lifetime of infected patient and totally increase symptomatic and abrupt of the disease. So infection of both viruses, hepatitis B and D virus, always increase a risk of death or early cirrhosis or hepatocellular carcinoma. Right now, I would like to tell you about hepatitis A virus. So on the picture, you can see, you can observe electron micrograph of hepatitis A virus in culture. Wait a second. Yeah. There is a structure of P coronaviruses, and you already know it. It has capsid, primary icosahedral capsid, very small, 27 nanometers, extremely small. Inside, you can observe single-strand positive sense RNA. The HAV genome has a VPG protein. The capsid is even more stable than other P coronaviruses to acid and other environmental factors. There is only one serotype of hepatitis A virus. So, very simple structure. Once again, naked capsid, single strain positive sense RNA. That's important things. In pathogenesis, one of the most common way, it's the oral consuming of the virus, and then it crosses intestines, come into the blood, come into the liver, replicate itself inside hepatocytes, destroy hepatocytes, come into the bile, and released with stool out of the body, so can infect another person. It's the short scheme of pathogenesis. Our mechanisms of defense, including as interferon, limits viral replication. Natural killer cells and cytotoxic C cells are required to eliminate infected cells. So our cells also can destroy hepatocytes and a bit increase the possibility of danger form of infection. But primary, it's not so important. It's just for prevent the replication. Antibody, complement, and antibody-dependent cellular toxicity also facilitated clearance of the virus and induction of immunopathology. So immunopathology is exactly when our immune system destroy our own cells. Destroyed because of cell already infected and will die anyway, and we have to destroy it because we can increase the permeability of the disease. Instead, resulting from the damage of the liver occurs when the cell-mediated immune response and antibody to virus can be detected. Antibody protection against reinfection is lifelong, so antibody will protect a person during all life after recovering. That's good news. Oops. So, in the structure of infection diseases, approximately 30% of all acute cases of hepatitis are caused by HAV. You know, virus spread very easily in the community because the most infected people are contagious 10 to 40 days before symptoms. Moreover, Up to 90% of infected children and up to 15% of infected adults have an inapparent but productive infection. What does it mean, inapparent but productive? Inapparent means symptomless infection. Productive means virus reproduce themselves inside hepatocytes and released outside. So a person with such infection, danger for other people, but have no any kind of symptoms. It became very dangerous because such kind of infection, type of infection, can be very dangerous for other people who are not infected, because infected person don't know about the disease, feel themselves absolutely fine, but transfer the produce new viruses and can be a source of infection. There is a map about geographical distribution of HIV. Red color is the when hepatitis A are prevalent. Of course, there is not totally and exactly how many percent, etc. It's just of detection of antibodies. You can see a lot of countries in South region which marked as red. Yeah, primary, a lot of developing countries, some developed, but primary developing countries. They deal here in two factors. First factor, lack of, inability to drink pure water for a lot of people, and environment, warm environment fit for virus spreading. Of course, the population also important, medicine also important, but anyway, two primary factors I described. So once again, I will talk right now about both hepatitis A and E because it's very, very similar. So capsid, very strong for environmental factor. Contagious periods extend from before to after symptoms. So a person, danger for surrounded person before symptomatic stage and after that. So anyway, can be easily transmitted. Asymptomatic cases, like I said, inapparent infection, very common for hepatitis A virus. Transmission, fecal oral route transmission only. Ingestion of contaminated food or water can cause infection. Understandable, I guess. HIV is shellfish from sewage contaminated water, also can be a source of infection. Virus can be transmitted by food handlers, their care workers, and children very easily. At risk, so people who are living in unsanitary areas, children with mild disease, a lot of asymptomatic cases, and daycare centers are major source of spread the hepatitis A infection. Adults with abrupt onset of hepatitis, and for pregnant women, high mortality associated with hepatitis E virus. So that's important fact. Hepatitis E virus very dangerous for pregnant women because can cause up to 20% of death. Geographically and seasonally, there is no actually seasonal incident and distributed worldwide everywhere. We can control hepatitis A virus by passive antibody protection for contact people. So if a person had a contact with infected one, we can try to protect them by passive immunization. Killed vaccine for hepatitis A virus available everywhere. Live vaccine for hepatitis E virus available in China, but not everywhere. World Health Organization didn't accept that. For laboratory diagnosis of HAV infection is generally made on the basis of the time, course, and clinical symptoms, the identification of the known infected source, and the most reliably the results yielded by specific serological test. The best way to demonstrate an acute HAV infection is by finding anti-HAV immunoglobulin M. Is measured by an ELISA test or radioimmunoassay. So please detect it. IgM here, IgM, very important to understand acute infection. So if the person has the symptoms of hepatitis, we can do simple ELISA test to finding IgM for hepatitis A virus. If we did that, we can be sure there is an acute infection of hepatitis A virus. If not, it's a question. Perhaps another infection agent or a problem cause the same symptoms, because the symptoms pretty similar for many different kind of infections and other problems with health. So isolation of the virus is not performed because efficient tissue culture systems for growing the virus are not available. I mean, we can cultivate virus, but not efficiently for the diagnosis. That's why we can't use it. But in generally, we can cultivate virus in the lab. So that's it about hepatitis A virus. And the next sub question, it's about hepatitis B virus. Look at the picture. There is a hepatitis B virus under electron microscope. Here you can see, please look here, that one. This is an infection party cell of hepatitis B, so real virus. Nearby, small one, small. It's an spherical virus which can't infect, and you can observe like rod shape. This one, another one here, a lot of. It's a specific surface antigen. So I will tell you about it once again, but right now, please just observe that and try to memorize how does viruses looks under the microscope. All of these structure, it's the parts of hepatitis B replication inside the cell. So HBV is the major member of hepatnoviruses, Hepatnoviridae family. Other members of this family include woodchuck, ground squirrel, and duck hepatitis viruses are not important for people. Virus has enveloped virion containing partially double-strand circular DNA genome. I will show you. Replication is through the circular RNA intermediate. HBV has a strict tissue tropism to the liver and can't infect in other cells. HBV infect the cells, produce and release a large amount of hepatitis B surface antigen particles lacking DNA. So, once again, hepatitis B surface antigen or HBS antigen, important thing. So, has no DNA totally. The HBV genome can integrate into the host chromosome. Look at the picture. There are all forms of hepatitis B viruses. So we can observe here Dane party cell, that one. Then small spherical structure and HBS antigen. That's important. HBS antigen, it's a part of supercapsid. Look here. Hepatitis B surface antigen. HBS antigen equal HBS antigen, what you can observe on the bottom. So, hepatitis B surface antigen located on the surface inside supercapsid. We also has two more antigens. HBC antigen means core antigen, HBE antigen, enveloped antigen. Both of them located in the capsid, but it's a different antigen. DNA inside, and it's double strained, but one strain shortly than another strain. Hepatitis B virus can cause acute or chronic infection, symptomatic or asymptomatic disease. Which of the occurs seems to determined by the person's immune response to the infection. So, if we can detect in the diagnosis both HBS antigen and HBE antigen components of the virion in the blood, indicate the existence of an ongoing active infection, primary acute stage. HBS antigen particles continue to be released into the blood even after virion release has ended and until the infection is resolved. So, HBS antigen, one of the most important in the diagnosis of the infection. But HBE antigen important for the diagnosis of current infection, acute infection. Two kinds of antigens. So, like conclusion about HB hepatitis, and as you see here, I already include here C and D viruses because there are a lot of similarities. So first, enveloped viruses, all of them, is labeled to drying. HBV virus is less sensitive to the detergents than other enveloped viruses. Virus is shed during asymptomatic period, important because a person can be dangerous. HBV cause up to 10% of chronic infection, as I told. HCV up to 70% of all cases cause chronic infection. Transmission for all these hepatitis, blood, semen, and vaginal secretion, but for hepatitis B, also saliva and mother milk, actually in big amount. Weird transfusion, needle sticks injury, shared drugs, needles, of course, sexual intercourse, any kind of sexual intercourse, and breastfeeding can be a way described as a way for transmission. But for hepatitis C virus, it's primary through the needles, through the injections. But for hepatitis B viruses, Also, it's like a 50-50 for needles and sexual intercourse. At risk. Who are at risk? For children, mild asymptomatic disease with establishment of chronic infection. So it's very important because for child, when the children have no any kind of infection, so but can be easily developing into chronic infection. Adult, insidious onset of hepatitis, and you know the symptoms. HBV infected people, co-infected or super-infected with hepatitis Z virus, abrupt, more severe symptoms with possible fulminant disease. I will explain about that, what does it mean, but please remember two things. Co-infection, when hepatitis B and D virus comes together, and superinfection, when person firstly get hepatitis B virus and after some time get hepatitis D virus, much more severe than co-infection. So super disease. Adults with chronic hepatitis B and C or C at high risk for cirrhosis and primary hepatocellular carcinoma. So again, there is no seasonal incident and found worldwide. We can control hepatitis B virus by hepatitis B vaccination, and we can control all diseases by screening of blood. So screening of blood critically important in resolution of epidemic incidents. Lab diagnosis. Initial diagnosis of hepatitis can be made on the basis of clinical symptoms and the presence of liver enzymes in the blood. Okay, we understand that and we know it's a main diagnosis if a person already has some symptoms. However, the serology of hepatitis B virus infection describes the course and the nature of the disease. Acute and chronic hepatitis B infection can be distinguished in the presence of HBs antigen and HBe antigen. So depend on a type of antigens which present in the blood. I will explain it a bit later. So antibodies to individual antigens also important in diagnosis because can tell us about stage of infection. HBs antigen and HBe antigen are secreted into the blood during viral replication. So both these antigens always present in blood during the infection. The detection of hepatitis E is the best correlate to the presence of infection virus because, as I said, hepatitis surface antigen can be present after the resolution of the infection, after the virus already destroyed and infection already treated, person recovered by antigen, surface antigen can be detected still. So a chronic infection can be distinguished by the continued finding of hepatitis E antigen, S antigen, or both, and lack of detectable antibody to these antigens. Antibody to S antigen indicates resolution of infection or vaccination. So in infected person, we will never find antibody to surface antigen. Antibody to core antigen, please look here, core antigen, HBC antigen, indicates current or prior infection by HBV, and immunoglobulin M to the same core antigen is the best way to diagnose a recent acute infection, especially during the period when neither HBs antigen nor anti-HBs can be detected. It's like a window for detection. Detection of antibodies to E antigen and S antigen is obscured during infection because the antibody is complexed with antigen in serum. So we have to distinguish between antigens and moreover between classes of antibodies, not just to say there is a presence of antibody to HBC. No, we have to understand, does it IgM or IgG? Let me explain it to you on the table. Laboratory diagnosis used serological markers, following serological markers. Let's talk about this table carefully, about 5-minute break. You can see this table, and 5 minutes later, I will explain what does it mean. Can you hear me?
Yes, sir.
Yes, sir.
Yes,
Excellent.
sir.
Yes, sir.
So let's continue. Serological markers. On the slide, you can observe two kinds of tables. Upper table with disease state, when the person are infected. We can understand it because infection virus here can be detected during all steps of the infection you can observe. So here you can plus means positive reaction for PCR, and we can detect DNA of virus all states. Antigens, surface antigen also can be detected during all steps, all stages. Another antigen, HBe antigen, doesn't detect it during early or pre-symptomatic stage, and also doesn't detect it in reconvalescent stage, when the late acute means a person start to resolve and after treatment, for example. So what's important here? HBe antigen important to understand acute stage or chronic stage of infection. But how to distinguish acute stage of infection from chronic stage of infection? We can describe it by only one antibody to core antigen, HBC antibody present in huge amount in chronic disease. But in acute stage, it also can be present. But only immunoglobulin class M in acute stage are present. But in chronic, both IgM and, of course, mainly IgG in huge amount. So when you understand by the lab diagnosis there is immunoglobulin G to core antigen, you can make a conclusion about chronization of the disease process. In the reconvalescent stage, also core antigens start to be present, but also a antibody to E antigen comes to present in the blood. In healthy state, when the person not infected, you can observe there is no virus, there is no antigens, but there are antibodies. Of course, core antibody and surface. That is a difference between vaccinated person, because vaccinated person has only anti-surface antibody. Anti-surface antigen antibody. So that's important, because if you would like to understand as if a person will show you a positive reaction for immunoglobulins to surface antigen, it's not means a person had the infection. Perhaps a person vaccinated. So have you any questions about lab diagnosis? Actually, it's a pretty tricky topic.
No, sir.
Okay.
No, sir.
Okay. So next subtopic, next chapter of today's lecture, hepatitis C virus. So it's enveloped virus with glycoproteins on the surfaces, which has core, means capsid, and RNA. Intermediate virus by the size, approximately 60 nanometers in diameter, and virus is spherical shape. So nothing totally special. But what is totally special? Special in this virus, in fact, as I already told, more than 117 million people over the world, and mainly such kind of patient also infected with hepatitis B virus or with human immunodeficiency virus, HIV. So Chronic hepatitis often leads to cirrhosis and potentially to hepatocellular carcinoma, so pretty danger bad infection. C virus is only member of the family Hepatitis is genus, and from the family Flaviviridae, we know this family already. It's 30 to 60 nanometer diameter, as I told you, and its positive sense RNA genome. Enveloped capsid, as I already told. Antigenic variability, very huge, makes development of the vaccine very difficult. So very changeable virus, a lot of strains. That's why immune system can't recognize all of them, and antibodies not protected for another strain than you vaccinated, for example. In pathogenesis, what's important? The ability of C virus to remain cell associated and prevent host cell death promotes persistent infection, but results in liver disease later in life. What does it mean? Such kind of virus can live very long in infected person without providing any kind of specific symptoms, but later it will come as a chronic infection immediately. So cell-mediated immune response are responsible for both the resolution of infection and tissue damage. So it's like both sides of the coin when we have positive and negative result of the immune response. Immunopathogenesis, important for all hepatitis and for C also. Antibody to hepatitis C virus is not protective. So it can be detected, but it's not means a person protected against the infection. Look here. There is a picture how does liver changed after the initial infection of C virus, and you can see there is there are several stages, and there is also hepatocellular carcinoma and liver cirrhosis stage. Both the stages are developed after many years. Look here, 10 to 40 years should be spent for this step. But anyway, it's not very long time if a person infected, for example, at 20, so 10 plus years, so 30, and primary hepatocellular carcinoma means death. But of course, not all people are surviving till this time. Maximum 5% of infected people surviving till hepatocellular carcinoma stage. But a lot of people has such symptoms as chronic hepatocytes and liver steatosis. So primary is transmitted with infected blood and secondary sexually. Intravenous drug users, tattoo recipients, transfusion and organ recipients are at a high risk for infection. Almost all, but actually more than 90% of HIV infected people who are intravenous drug users are infected with hepatitis C virus. Please understand, that's not mean all people who has HIV infected also with HCV. No, intravenous drug users who has HIV infection also has HCV. That's it. So the high incidence of the chronic asymptomatic infection promotes to the spread the virus in the population. As I always talking, asymptomatic carriers, asymptomatic person much more danger in epidemiological case for people than person with symptoms, because a person without symptoms don't know about it usually, and usually never care about the protection. Of course, not self-protection, but the protection for surrounded people, for friends, family, doesn't matter, for any people. So I took a picture about United States because there is the most detectable. A lot of people, more than 100,000 people, results taking in this graph. So very accurate, but for United States, of course. Look here, how they got the infection. The big piece of pie, 60%, infection drug use, so intravenous drug users. Only 15 sexually. Transfusion before screening, 10. But please understand, right now, a lot of blood checked for the screening. It's under law transfusion or transfusion in other countries, or transfusion before screening program, because the graphs take more than 30 years data. Lab diagnosis. Again, ELISA test widely used to recognize antibody to antigens, to hepatitis C virus antigens. Also, we can use PCR to detect genome, so RNA genome. Seroconversion occurs within 7 to 31 weeks of infection. It's important because seroconversion is the window time when a person already infected but have no antibodies in detectable amount. ELISA test is used for screening the blood supply from normal donors. So how to detect genome? Of course, PCR reaction, but also quantitation by real-time PCR, widely used right now. It's not only helps to detect how many viruses inside blood, so is it strong contamination or not, but also helps to monitoring the antiviral drug therapy, because a person who getting the treatment must understand, does treatment effective or not. How to understand? Only count how many viruses in blood. If there are not many viruses, treatment is effective. If there are many viruses, we're talking about bad treatment strategy. Genetic assays are less strain specific and can detect hepatitis C virus RNA in seronegative people. So what does it mean? If do you remember I told seroconversion 7 to 31 week. This window means have no a lot of antibody, but can be detected RNA during this time. So if a person are negative in ELISA test, we can detect RNA by PCR technology and provide the proper result. But ELISA test much more cheap than PCR and much more simple, of course. So G virus, shortly about G virus, also enveloped, also has a capsid protein, nucleic acid, RNA, and of course antigens on the surface. G virus, also known as hepatitis B virus C, GBVC. It's because it's very similar to hepatitis C virus in many ways. D virus is also flavivirus, is transmitted by the same way as hepatitis C virus, and a predilection for chronic hepatitis infection also. It is identified by detection of the genome by real-time PCR or other RNA detection methods, but not by ELISA test, for example. That's it. So, and hepatitis D virus, D virus and B virus, always together. D virus can't replicate itself without B virus. Look at the structure. Hepatitis D virus here, and you can observe hepatitis D virus has same antigens on the surface as hepatitis B virus. Absolutely. But what's the problem? Hepatitis D virus can't produce, so can't synthesize these antigens by itself. So hepatitis D virus need a factory of surface antigens, and hepatitis B virus in this case is exactly a factory. So approximately 15 million people in the world are infected with Delta virus, D. And the virus is responsible for causing 40% of fulminant hepatitis infections. So amazing percentage. D virus is unique in that it uses hepatitis B and targets cell protein to replicate and produce its one protein. It's a viral parasite, and actually you can see even fleas have fleas. So virus which parasite of the virus, but it's not exactly correct because hepatitis B virus no need the surface antigens in big amount because it already overproduce it. So hepatitis B surface antigen is essential for packaging the virus. Without this antigen, packaging never happens and attachment also not. So, virion of D virus, coat of them totally made by hepatitis B surface antigen, but inside single-strand RNA genome and specific delta antigen in the core, but it's not exactly the core, it's a primary capsid. Lab diagnosis. Also, we can primarily detect RNA of this virus by PCR, of course, the delta antigen or anti-HDV antibodies. So specific antibodies produced for specific delta antigen, what I already showed. ELISA and radioimmunoassay procedure are available for detection. Delta antigen can be detected by these methods inside the blood during acute phase of disease in a detergent-treated serum sample. Real-time PCR can be used to detect the virion genome in blood during all stages of infection. Last one for today, hepatitis E virus. There is a model of hepatitis E virus, how it looks like on the 3D model. Hepatitis E virus, or sometimes we can call it E non-A non-B hepatitis. We call it E because it's enteric or epidemic, but primary enteric. It's predominantly spread by fecal-oral route, especially in contaminated water and sewage water. hepatitis E virus is unique but resembles the calciviruses based on its size and structure. Actually very small also as hepatitis A virus. HEV is found through the world and it's most problematic in India, Pakistan, Nepal, Burma, North Africa, and Mexico, because the regions with very problematic situation with pure water for drinking. The mortality is rare, is 1% to 2% of all cases. Oh, sorry, mortality not rare, of course, approximately 10 times higher than associated with HAV disease, so much more danger, and especially serious in pregnant women. Mortality rate approximately 20% of all infected. So if the pregnant women infected by hepatitis E virus, it's very bad situation. So that's it for today. Take care. If you have any questions, stay here. If you have no any questions, you can be free. Hope the lecture was helpful for you.